Good afternoon, ladies and gentlemen, and welcome to the last session of this scientific forum, Age Open Science. We would like to thank all of you for joining us today. We still have a relevant and interesting topic about aging and longevity. And we would like to welcome also the audience who are following this session by streaming. And we are starting calling to the stage Marta Trujillo, who is a professor of microbiology. Thank you very much, Marta.
Thank you very much. Good morning to everybody. It's wonderful to be sharing this session with you in this beautiful site at Fonseca College. So it is my privilege to present Professor Steve Horvath from the David Heben School of Medicine at the University of California in Los Angeles. Among other subjects, Dr. Horvath's area of research lies on the interaction of biostatistics, bioinformatics, computational biology, and cancer research. His team applies these tools to a wide spectrum of diseases, including Alzheimer's and early menopause. Today, Steve will present his most recent studies on DNA methylation of aging in different mammals. Therefore, I invite you to the stage.
Here. It's such a privilege to be here. I think Salamanca is really the most beautiful city on earth. In California, we also have beautiful architecture like here, but was all built in the 1920s. And you have the original building, so I love that. I will talk today about aging clocks, how to measure aging, aging in different organs, organismal age, and it's all based on properties of the DNA molecules. So chemical changes known as epigenetic changes of the DNA molecule. You can think of it a bit like rust. You have DNA, and as we age, rust accumulates. And by tracking the amount of methylation, we can measure age quite accurately. My personal scientific interest is to measure aging in people who follow the perfect lifestyle. I want to measure innate aging processes. So as an example, you have a person who sleeps enough, they eat vegetables, they don't smoke, but something still ages in them, and I'm interested why do they age and how to measure it. I give you here an example of a bodybuilder at age 71, and according to many clinical biomarkers like grip strength and walking speed and other markers of frailty, this man would look very young. But this is, of course, deceptive because he cannot fool molecular markers. So, my interest is very much in molecular biomarkers. And many people talk about biologic age. Everybody has an intuitive feeling what is biologic age as opposed to chronological age. But how to measure it is actually very difficult. And there are different approaches to measuring biologic age. One is described on the right-hand side of this PowerPoint slide, which is clinical measures of pathology, frailty, muscle weakness, lung dysfunction, memory function. For example, Cynthia Kenyon just talked about physiologic age. So all of that goes in that direction of pathology. On the upper square, you see measures of tissue pathology. A pathologist can keep track of aging in your tissues. And in my talk, we will talk about genomic data. There are many different types of genomic data: transcriptomic data, proteomic data, metabolomic data. In this talk, I will focus on methylation data. But I want to give you this overview because the literature has come up with many, many different ways of measuring biologic age, and they can be broadly distinguished by these domains of variables. Now there's one variable that is really quintessential, which is chronologic age. If you have a biomarker that does not correlate very strongly with chronologic age, then it's really not a biologic age measure. Rather, it's maybe a measure of clinical pathology, but not a biomarker of what I call innate aging. Remember, I'm interested in innate aging processes that actually start very early on, even in children. So you will hear me talk about clocks, and I want to tell you that in this talk, the word clock really refers to a number. There's an algorithm, there's a software that allows you to arrive at a number. And that number either measures chronologic age or, more importantly, risk of mortality, morbidity. And because there are so many different biomarkers of biologic age, I've developed 4 axioms that I feel a true biomarker of innate aging has to satisfy. And the first is that it has to be a quantitative measure, a number, which is highly correlated with chronologic age. Otherwise, it's not an aging marker. The second property is a very hard hurdle for most biomarkers, which is it really should apply to all mammalian species. And the reason is all species age. So it would be nice to have a biomarker that applies to all of these species. The third axiom is that this biomarker should relate to mortality risk. So it should predict lifespan. But at the same time, we need to acknowledge that it is different from a mortality risk estimator. And why is that? Because you can die in a million different ways. You can get an infection and be dead in a week. You can take a poison, you're dead in a minute. So not everything that kills you ages you, so it's important to make that distinction. And the fourth property is, and the fourth axiom is, that your biomarker of age should relate to a fundamental property of most cells. And why is that? Because the intuition is most cells age. And also, we would like that these biomarkers we develop apply to cells growing in a dish so that we can identify novel anti-aging interventions. For example, Vera Gorbunova earlier today described wonderful experiments with fibroblasts or other cells. And so we need biomarkers that apply to these cells. So these are the 4 axioms that I impose on all true biomarkers of innate aging. And one of the biomarkers that satisfies these 4 axioms is DNA methylation. As I said, it's a chemical modification, typically at one of the letters of the DNA. The DNA has 4 letters, A, C, T, G, and the letter C sometimes get modified because a methyl group attaches to it. So again, I think of it like rusting. But conversely, in aging, the opposite also happens. Certain parts that should be methylated lose methylation. And DNA methylation is absolutely essential for development. A zygote will never differentiate into different cells. An embryo will never develop unless you have methylation. However, we now recognize that methylation also plays a role in the aging process. And coming back to these biomarkers of aging, DNA methylation allows one to build biomarkers with truly unique properties. And the first property is that they apply to the entire life course. They are not aging biomarkers, they are life course biomarkers, meaning we apply these to children, we applied these to prenatal samples. It's truly remarkable that these biomarkers link aging to development. And you will hear me talk a lot about that connection between development and aging. which is one of the big picture insights that we have gained through methylation. The second property of methylation biomarkers is that you can build what we call pan-tissue clocks that apply to all cells that have nuclei, all cells that have DNA. You can give me DNA from any source, I can tell you the age, you know. For example, what tissues have DNA? Saliva or buccal swab, what you do during the COVID test has DNA, I can measure your age. A blood sample, a nasal swab, a skin sample, urine. If you pee in a cup and you give me your urine, I'll give you your age very accurately. So anything that contains DNA. Now the third property is, in my opinion, the most remarkable property of methylation, which is you can build so-called pan-mammalian clocks. These are now clocks that apply to all mammalian species. It's one mathematical algorithm, one set of cytosines, one formula, and you can measure aging in all these species. So we distinguish different generations of clocks. The first generation clocks were developed in 2011. They measure chronologic age. The second generation clocks predict mortality risk in humans. Such clocks are actually being investigated in the context of life insurance companies. They want a buccal sample or a blood sample to assess your mortality risk, to price life insurance policies. But we, of course, use such clocks in human clinical trials. Now, the third generation clocks are these, what I call multispecies clocks. These are clocks that apply to different species, mouse and human, mouse and dog, and so on. So I will start with an example which 10 years ago I thought was impossible to solve, which is develop a clock for primates. And just to give you some context, about 60 million years ago, planet Earth was hit by an asteroid which killed all the dinosaurs. And 5 million years later, or so, or 10 million years later, we already had all the first ancestors of primates. And our species is separated from lemurs in Madagascar by 55 million years of evolution, huge differences, which leads to differences in DNA sequence. And so in principle, one would think it's very hard to build an epigenetic clock that applies to all primates. To solve this problem, we had to develop a new technology for measuring methylation, which is resistant or impervious to mutations. So here in the upper row, you see a DNA sequence that measures cytosine methylation in humans, and you can appreciate in all the other species you see mutations. But we have a mammalian array that is resistant to these mutations and can measure methylation. And we used then this array to measure methylation in 37 different primate species. Any primate that is interesting to biomedical research, rhesus macaque, baboons, vervet monkey, marmosets, but also lemurs, for example. And here I show you clocks, one clock, it's called the pan-primate clock, that is so accurate that if you're a statistician, you will say, well, you are overfitting. You will say this is so-called training set estimate. But I can assure you this is an unbiased evaluation of the clock. So, and in the middle panel, you see the application to humans, and the correlation is 0.98. I mean, this is a clock that applies to all primates and is particularly accurate, of course, in humans. Now, you can ask a more difficult question. take a pig. Let's build a clock that applies both to pigs and also to humans. And this is a challenging exercise because pigs live shorter lifespans. The maximum lifespan of a pig is only 23 years. Especially here because of the jambon. You will butcher them very early, you know, but which is delicious, I want to mention. But anyways, the oldest pig that has ever been observed was 23 years. But now the oldest human was 122 years. And in order to align these species, we use a mathematical trick. We take the age of the animal and we divide it by the maximum lifespan of the animal. And that ratio we call relative age. So it's a number between 0 and 1. It's a very naive way to align the species. You can think of biologically more meaningful ways to do it. But the interesting thing, though, is even this naive way of just this ratio allows you to build this multi-species clock that estimate relative age. And the middle panel shows you on the x-axis relative age, and on the y-axis the estimate based on methylation. And the green dots are human tissue samples, and the other color dots are pig samples, different tissues. And so you get this very accurate clocks for human and pigs. So why do we develop it? Well, because the mini pig is a medically very interesting model organism. For example, if you want to study transplantation and so on, and we now can measure aging using the same clock, both in pigs and in humans, thereby hopefully increasing the probabilities that things translate. Let's say you have a rejuvenation protocol, the Yamanaka OSKM rejuvenation, and it rejuvenates, for example, pig organs, then the chances would be increased that this will also apply to humans if you use this clock. I want to come to an example in sheep. So we built a human sheep clock, and you would ask why. And the reason was we wanted to evaluate the effect of castration. And it turns out people often castrate male sheep rams because that improves the meat quality. And so then we applied our human sheep clock to the ear samples of these castrated sheep. And the blue samples here show you that yes, castration actually decreased the epigenetic age of the ear samples of these males. And I think many men will now say, I also want to have young ears, you know. And but I want to caution you, we also evaluated the effect of castration in blood, and the results were disappointing. So castration does not affect the epigenetic age of blood. And we looked at it in dogs, we looked at it in cats, we looked at it in horses. So blood is different, which makes sense because blood cells have fewer hormone receptors. I now want to come to this question, can one build epigenetic clocks that apply to all mammalian species? And here I show you that yes, it is possible. And here we evaluate over 300 different mammalian species. On the right-hand side, you see again our predicted value of relative age. The y-axis shows you the predicted value. Correlation 0.95. So to me, this is truly a miracle of cytosine methylation. But it also reflects the technology that we use. Remember, we use a mammalian array that looks at highly conserved stretches of DNA. And that made it feasible. And it turns out it actually made it easy. So as long as you look at these highly conserved regions, it is actually straightforward to build a pan-mammalian clock. Here I show you how this, we call it universal pan-mammalian clock, applies to different species. The upper left shows you the human species, and you notice the clock tracks again development. Carefully look, age 0 all the way to over 100. So it's a life course clock. And the upper middle panel shows you how the pan-mammalian clock works in mice. Then you see dolphins, then you see dogs, then you see elephants, then you see bats. It's just remarkable that it's one mathematical algorithm. It's about 600 cytosines, and it just works really well. Several authors or speakers have talked about stem cells, Juan Carlos Belmonte, Henry Jaspers, and many others. Yes, epigenetic clocks indicate that iPS cells and stem cells are perfectly young, negative ages. Here I show you a time course experiment, 45 days of reprogramming in the dish, and the blue line shows you how the epigenetic age gradually decreases. But you really see these drastic decreases actually only after about a week, about 10 days, then you really pick up this effect. Initially, it's rather weak. But now you could ask the question, well, what about iPS cells in different mammalian species? Do they also show prenatal ages? And here I show you collaboration with Vera Gorbunova and Andrei Selyuanov, where we've shown that the iPS cells from the naked mole rat, again, are perfectly young. But one side story of this study was that we do have very accurate clocks for the naked mole rat. And that is interesting in its own way, because the naked mole rat has negligible senescence on the phenotypic level. It doesn't seem to age. But when you look at the methylome, there's no question it ages. It actually already ages in the first 5 years, So in my interpretation, the naked mole rat is resistant to these changes, And there's another long-lived species that is quite resistant to these changes, which is humans, We are also remarkably long-lived, and clearly you can measure our age. Oh, sorry. We evaluated several other species in collaboration with Vadim Gladyshev, goats, pigs, bats, and in many of these species we also see that the iPS cells are perfectly young. I wouldn't say this question is settled yet. We need to profile many more species, but I would say for many species we observe that. I want to talk about what I call gold standard perturbations that affect the epigenetic clock in the expected way. Half the people here talked about caloric restriction, and there is no question that has a very strong effect on the epigenetic clock, especially in liver. Then Cynthia Kenyon talked about these very interesting mice, the growth hormone receptor knockout mice, these dwarf mice, where you dial down IGF-1. These mice live much longer, and to us, this is one of these gold standard interventions that affects the epigenetic clocks in multiple organs, and certainly liver, kidney, but perhaps even brain. The other gold standard intervention that was talked about by another speaker, Tom Rando, is peribiosis. And I'll talk about it in a second, but that has a very strong effect on rejuvenating the clocks. If you connect an old mouse to a young mouse, you get the young blood that rejuvenates the organs. Now, on the flip side, how do you age a mouse? High-fat diet. You make the mouse obese, very strong effect in liver. And trisomy 21, which is also known as Down syndrome. An extra copy of that chromosome very much affects the age in multiple tissues. So here I show you the Peribiosis experiment, where an old mouse is connected to a young mouse. And the left panel shows you how the rejuvenation, so of the epigenetic age, according to our Panmammalian clock. But I could have taken any clock. We actually have many, many clocks. Any clock would show it. So yes, if you get the circulation of a young animal, many organs get rejuvenated, and the striking thing is that even can be observed 3 months after you disconnected the mice. So this effect persists. And conversely, Vadim Gladyshev and Bohan Zhang have shown that the opposite is also true. So the young mouse connected to an old mouse experiences increased epigenetic age in multiple organs, even 3 months after they were disconnected. I want to talk about accelerated aging diseases, progeria. There are 3 progeria where I'm very confident that they accelerate the epigenetic clock. The first one I mentioned, Down syndrome, no question, because we've validated this over and over. The second is Werner's syndrome. The limitation is we only studied that in blood, but there was a very strong effect. The third is cocaine syndrome, where an Italian team has shown that fibroblasts show accelerated epigenetic aging. So to me, these are the top 3 progeria that I'm quite confident in. Now here I now move to the mice. What I just talked about is human Down syndrome, but remember we have Panmammalian clocks. So here we studied a model of Down syndrome where 2/3 of the relevant human gene region were moved into the mouse. And here you see 3 months later, so these are young mice, 3-month-old mice, you already observe epigenetic age acceleration in blood, cortex, liver, matching the human results. To me, this is kind of the future of aging studies. You use a biomarker that applies to multiple species, and therefore you can readily move insights gained in one model organism to that of another. I do want to mention another progeria, and that's arguably the most famous progeria that, for example, Juan Carlos Belmonte talked about, Hutchinson-Gilford progeria, which afflicts children. It's heartbreaking. And, but the evidence was ambiguous. So we find fibroblasts of these children exhibited faster aging, but when we looked at blood samples from these children, we did not see epigenetic aging effects. And this is another paradigm for epigenetic clocks, because often you observe rejuvenation in one tissue but not the other. I mentioned earlier castration. You see it in skin cells, but you don't see it in blood cells. Now I want to come to a different topic. Forget age or calendar age. I now want to talk about, again, maximum lifespan, which is a species characteristic. Vera Gorbunova mentioned the bowhead whale, which is the longest lived mammal, can live up to 211 years. On the other end of the spectrum, you have mice and shrews, very short-lived species. And you can ask the question, can I predict the maximum lifespan of a species based on cytosine methylation? And here I show you the results on a log scale. The y-axis is the predicted maximum lifespan. Again, these are cross-validation estimates, and you see a correlation of 0.89. It's really, clearly a smoking gun that methylation plays an important role for maximum lifespan. And I want to briefly mention an outlier. disappointing results were actually humans. The one species where our predictor doesn't work well is humans. So humans live much longer than what the methylation predictor would infer. And but we don't stop there. We, of course, want to find the individual locations on the DNA that strongly relate to lifespan. And so these are now the cytosines on the DNA. And when you carefully look at the gene names, you will find many Hox genes. And Cynthia mentioned earlier today how important Hox genes are for patterning, for development. But these Hox genes are our top hits for maximum lifespan. It comes back to what I told you earlier. The biggest insight, or the overarching insights from methylation studies, is this close connection between development and aging. And here I just show you a bioinformatic enrichment analysis of these top cytosines that relate to maximum lifespan. I mentioned Hox genes. More generally, genes that play a role in organ development. But I also found that there are certain human traits that overlap. So obesity, interestingly, so genes that relate to human obesity, they overlap with genes found in studies of maximum lifespan. And another curious finding is genes that relate to our human biomarkers of mortality risk. Remember, I told you we have biomarkers that predict mortality. One of our best ones is called GRIM Age, GRIM named after the Grim Reaper. And so it predicts human mortality, but the genes that were found in genome-wide association studies of GRIM Age, they overlap with these maximum lifespan genes. And many people always ask the question, well, which hallmarks of aging relate to epigenetic clocks? And 10 years ago, we didn't have an answer. But through very careful studies by many people in the field, I show here a paper from Ken Raj, we have a pretty good understanding by now. So here are famous hallmarks, and the first three do not relate, to our disappointment. So Manuel Serrano and others talked about cellular senescence, the important role of senolytics. So for example, Pura talked about it on the first day. But you know, we never saw a connection between cellular senescence and epigenetic clocks. And relatedly, we don't really see a relationship between telomere shortening. Another important hallmark is genomic instability, and you can induce it through radiation. And again, radiation does not seem to accelerate epigenetic clocks. Conversely, though, 4 other important hallmarks relate. First of all, nutrient sensing. I mentioned to you this insight of perhaps obesity relates. Mitochondrial dysfunction relates to epigenetic clocks. Stem cell exhaustion, more generally, stem cell properties very much relate to epigenetic clocks. And what is known as cell-cell communication. Remember, I talked to you about peribiosis and really young plasma. And so that very much relates to epigenetic clocks as well. So I want to finish with humans. So here I show you the aging trajectory across the lifespan, the blue line. And if we can measure your age, you give us a blood sample, I can tell you you age a bit faster, you age a bit slower. And yes, if you age faster, then you have a slightly elevated risk, slightly that you are at a higher risk of mortality. And so we look at this discrepancy. So Cynthia called this discrepancy from the blue line delta age, but the discrepancy between the estimated age and your chronologic age. And you see on the lower level how this discrepancy relates to body mass index in the liver. And we find that, yes, people with fatty liver age much faster. It's an obvious insight. I mentioned our mortality risk predictor named after the Grim Reaper. And if you're in the top 5% of fastest agers, then your mortality risk is more than 2 times that of an average person. And what do I mean by that? Let's say you get up in the morning and compare your mortality risk on that day to that of the average person. So yes, if your grim age is very high, then you have double that rate. It's still low, right? Everybody has a very low risk of dropping dead in the morning. But anyways, so yeah, in conclusion, we have epigenetic clocks that link to these 4 hallmarks of aging: stem cell exhaustion, nutrient sensing, mitochondria, intercellular communication. Epigenetic age is distinct from telomere shortening and markers of senescence. And what does that tell you? Epigenetic clocks are not the whole story, you know. So if you design clinical trials, you really need to measure many, many biomarkers of aging. Epigenetic clocks are, in my opinion, an important biomarker. They should be added, but it would be foolish to focus on them exclusively. I did mention you can use cytosine methylation to build Panmammalian clocks. You can use cytosine methylation to estimate the maximum lifespan of species. We have epigenetic clocks for any model organism you can think of, if it's a mammal. And I started a non-profit foundation, so if you're interested, send me an email, we measure the ages. Epigenetic clocks are widely used in preclinical studies, but also increasingly in clinical studies. And I stop with acknowledging my many collaborators. Thank you so much.
Thank you so much, Steve. Now Marta Trujillo will introduce the following speaker. Thank you, Marta.
So hello again. So it is now a great honor for me to introduce María Blasco, who is the director of the Spanish National Cancer Research Center. Dr. Blasco's research is focused on understanding why cancer occurs and demonstrating the role of telomeres and telomerase in this process. She also does a great job of disseminating the importance of science and research in our society. María Blasco is the current president of SOMA, the Alliance of Severo Ochoa Centers, and among other awards, she received in 2010 the Santiago Ramón y Cajal Prize for Research in Biology. This morning, Maria will talk about the activation of telomerase of treatment for telomere loss disorders. So it's all yours, Maria.
Hello, thank you very much. So apologies to the Spanish speaking. I was told that it's better that I speak in English, so I will speak in English. So we are interested in understanding aging to be able to cure diseases. And one of the molecular causes of aging is the attrition of the ends of our chromosomes that are called telomeres. An analogy has been made between the telomeres and the plastic at the end of a shoelace. So these telomeres shorten associated to aging, but there is an enzymatic activity known as telomerase that is able to elongate telomeres. And thanks to telomerase, we are able to maintain a species. So independently of the telomere length of the father and the mother, the new individual will reset telomeres thanks to telomerase and have, hopefully, a normal life. But telomerase is silenced when we are born, and this leads to telomere shortening with aging. And a proof of that is that there are humans that have mutations in telomerase and other telomere genes, and they will age prematurely, they will develop prematurely a number of diseases that are known as telomere syndromes. Also, individuals that are not mutant for telomerase, they can have a faster telomere aging, and this has been associated with a higher risk of different types of diseases. Not only in humans, telomeres are important, also in mice. Actually, we contributed to generate the first telomerase deficient mice, and this was a demonstration that when telomeres were short, this was sufficient to cause aging. And already in the first generation, these telomerase knockout mice have a phenotype. They have a shorter median and maximum lifespan, suggesting that telomere length is also important for mouse aging. We more recently studied telomere length in different species of birds and mammals. We did this in collaboration with the Zoo of Madrid and with a group working in the University of Barcelona, studying the Audouin skull. And what we did is to measure the rates of telomere shortening in different species, and we saw that the telomere length did not correlate with longevity, but we saw that the rate of telomere shortening showed a very good correlation with median lifespan and with maximum lifespan, and we could adjust the rates of telomere shortening to a power law so that we could predict longevity only by knowing the rate of telomere shortening. So you can have, for instance, elephants and flamingos. They are very distant in evolution, but they share the same rate of telomere shortening, and they also share a similar longevity. On the other hand, mice, for instance, you can see that mouse shorten their telomeres 100 times faster than humans. So even though they are born with long telomeres, their telomeres are shortened much faster. So the interest of my lab has been to use telomerase as a way to delay aging and to maybe cure age-related diseases. So in our first experiment, we showed that if we generate mice that have longer telomeres, and these were transgenic mice, we could make these mice live longer. We could also extend their health span. And you can see here 2 mice of the same age. The one that has longer telomeres really looks like a younger mice. So we showed that telomerase was able to delay many different pathologies of aging, and because of that, it could also increase longevity. More recently, we generated telomerase gene therapy. This is a type of advanced therapy that consists in activating telomerase in mice. And we use adeno-associated viruses. And because we don't have telomerase in the adult organism, we hope that if we could activate telomerase, maybe this would delay aging. This would be sufficient to delay aging. And these vectors are non-integrative, so telomerase will be diluted out as the cells proliferate. And by using this strategy, we treated middle-aged mice and all mice with a single injection with these vectors. And we could see that the mice treated with telomerase show longer telomeres and less DNA damage as they age. And we could delay not one, but many different pathologies associated with aging. Also, cancer was delayed, supporting that cancer is also an age-associated disease. And we could increase the survival of these mice also by 24% in the case of 1-year-old group and 13% in the case of the 2-year-old group. We have also shown that telomeres can increase longevity without activating telomerase. And we did this using an epigenetic trick, if you want. We did this by knowing that telomerase normally elongates telomeres in pluripotent cells in the embryo, but that we could also generate these pluripotent cells using the famous reprogramming. So in my group, we show for the first time that when you reprogram cells, the telomeres are elongated, but we also noticed that these telomeres keep elongating beyond the natural species of the inner cell mass in the blastocyst, and that we could generate embryonic cells with really what we call hyper-long telomeres. And we use these cells now to generate chimeric mice that are going to be formed by cells that come from embryonic cells with hyper-long telomeres. So these mice have longer telomeres when they are born. And we published a couple of papers, I'm just going to refer to the last one, in which we managed to generate 100% chimera. So 100% of the cells have already longer telomeres than normal when these mice are born. And we noticed some interesting features of these mice. They had a lower body weight, and it was because they accumulate less fat as they age. We saw an improved glucose metabolism as these mice age. So these mice had a better glucose metabolism. These mice had longer telomeres as they age, and they accumulated less DNA damage and less markers and less telomere damage. And I didn't show that here, but they also have an improved mitochondrial function. So either if we activate telomerase or we generate these mice with longer telomeres by using this reprogramming trick, this is showing that long telomeres are delaying aging and improving, let's say, the health state of mice. So what we are doing now is that we have tried to see whether this telomere gene therapy could be useful for the treatment of age-related diseases. We started this as part of an innovation program with Roche, and we have shown that telomerase activation using gene therapy has therapeutic effects in mouse models of heart infarct, also in a mouse model of aplastic anemia that we developed in our lab, that is aplastic anemia triggered by short telomeres. And more recently, we have shown also a therapeutic effect in mouse models of pulmonary fibrosis associated to short telomeres that we have generated in the lab. And I will be referring to this in my following slide. So pulmonary fibrosis is the most prevalent telomere-related disease, telomere syndrome. And this was known because families with pulmonary fibrosis, they have mutations in telomerase and telomere genes. But even in the sporadic cases of pulmonary fibrosis, approximately 50% of the sporadic cases have been also associated to short telomeres. So short telomeres seem to have really a causal role in a high percentage of pulmonary fibrosis cases. So we think that short telomeres probably are synergizing with damage to the lung, and this is generating the fibrotic phenotype. The current treatments for pulmonary fibrosis are blocking the fibrotic process, but they are not curing the patients, and we think this is because you still have the short telomeres, and short telomeres are known to impede the ability of stem cells to regenerate tissue. This has been shown in many different tissue types by my group and other groups. So what we did next was to try to understand which is the important cell type for this role of telomeres in the lung. And we did this by inducing telomere dysfunction in different cell types. And we found that is the alveolar type 2 cells, the ones that are where it is important, the telomere function. So in other cell types, like the basal cells or the fibroblasts or the Clara cells, you don't get interstitial fibrosis. Only when you when you induce telomere dysfunction in alveolar type 2 cells, you have pulmonary fibrosis. So this told us that if we want to cure pulmonary fibrosis based on telomerase activation, we have to target the alveolar type 2 cells. So we generated a mouse model in which we generated, they develop pulmonary fibrosis because they have a combination of short telomeres plus an agent, a damaging agent to the lung. So we use a small dose of bleomycin that doesn't induce fibrosis in a wild-type mouse, but in the context of short telomeres, these mice develop pulmonary fibrosis. And we treat these mice with the telomerase gene therapy that is targeting these alveolar type 2 cells. And what we see is that the mice that were diagnosed with fibrosis, but we treated with the empty vector, they develop very severe fibrosis in the lung, as you can see here. But the mice that we treated with telomerase, we were able really to stop or to reverse the fibrosis. Half of them really don't show any fibrotic patches, and the other half, they only have small fibrotic patches. So it seems to be working, targeting alveolar type 2 cells with telomerase to prevent the progression of this disease. At the molecular level, we see that already 3 weeks post-treatment, there is less DNA damage, less markers of senescence and apoptosis. And if we isolate the alveolar type 2 cells from these mice, we see that we activate their telomerase, and we see also a downregulation of the DNA damage response and the immune signaling pathways, indicating that by targeting the short telomeres in this model, we are able to reverse different hallmarks of pulmonary fibrosis, both molecular and cellular hallmarks of fibrosis. So, of course, you can worry about maybe telomerase could be delaying or stopping the progression of fibrosis or even curing fibrosis, but maybe down the line you can have a tumor because telomerase could be synergizing with maybe oncogenic mutations to induce cancer. And in order to address this, we did an experiment in which we activated telomerase, and at the same time, we put an oncogene, a KRAS oncogene in the lung. And we waited until these mice developed lung cancer. Telomerase was activated at the time of the oncogene or weeks before the activation of the oncogene. And the results were that telomerase treatment did not change the natural history of these tumors. it did not make an impact on the number of mice with tumors, the tumors per mice, or the size of the tumors, suggesting that at least in the mouse and in this model, we could not see that telomerase was favoring tumor development in the lung. Therefore, we think that there is value in trying to activate telomerase in the lung for the treatment of pulmonary fibrosis. Another disease that we have been interested in is kidney failure, which is a prevalent disease. It's very similar to the lung fibrosis in terms of deposits of extracellular matrix, inflammatory cells, activation of myofibroblasts in the kidney. And recently, we have developed also a mouse model that induces kidney fibrosis due to short telomeres. So basically what we have done is to use a low dose of folic acid, that is a damaging agent to the kidney, which does not induce kidney fibrosis in a wild-type mouse, but in a telomerase deficient mouse model with short telomeres is sufficient to induce fibrosis in the kidney. You can see this by creatinine levels and looking at the blood urea nitrogen levels. Only the mice with the short telomeres are developing the kidney dysfunction, and actually this kidney dysfunction is actually kidney fibrosis, as you can see here by activation of myofibroblasts, collagen deposits. You can see look at the expression of all the collagens. So really, this is showing that short telomeres can be also at the origin of a disease like kidney fibrosis, at least in these mouse models. So this is the summary. We also found, I don't have time to tell you, we also find that short telomeres are triggering an epithelial to mesenchymal transition in the kidney, which is an important feature also of this disease. So due to this finding, we think we have started generating human vectors for telomerase to be able to target telomerase to the lungs and to the kidneys, and a new company has been generated from the CNIO and the Universitat Autònoma de Barcelona, which is called Telomere Therapeutics, that is trying to bring this new therapy to the patients. And to finish just my last 2 minutes, I would like to mention COVID-19. I was very intrigued when I learned that the SARS-CoV-2 virus infects alveolar type 2 cells in the lung, and one of the sequels is pulmonary fibrosis. And it also transduces the kidney, and one of the sequels is kidney dysfunction and kidney fibrosis. And this was very, it reminded me a lot of the telomere syndromes and the phenotypes associated to short telomeres. So we did a first study. This was a very simple study. It was very difficult to get samples from patients in Spain when we were in the middle of the pandemics. But we managed to get blood samples from a field hospital in Madrid that doesn't exist anymore. It was done for the COVID situation. And we just measured telomeres in blood, and we saw there was a correlation between the severity of the disease and telomere length in blood. More recently, we have gotten samples from the lungs of patients that had COVID, and we could confirm shorter telomeres in the COVID patients in the lungs, and also, of course, markers of fibrosis. This is already known, like Mason trichome is increased, collagen deposits are increased, activation of myofibroblasts are increased in the lung, and we saw that there is a correlation between the fibrotic markers and the telomere length. So the shorter the telomeres in the COVID patients, the more Mason trichome staining, and the more collagen deposits, the more activation of the myofibroblasts, and the less alveolar type 2 cells. So this is just a correlation, but it's suggesting that maybe short telomeres could be contributing also to the COVID-19 disease, and these patients could also maybe benefit from technologies that activate telomerase activity. So I will finish here by saying that we think telomeres are important players of the aging process. And finally, the people working in my group, I would like to particularly mention Paula Martinez for all the pulmonary fibrosis work, and the rest of the people of the lab. Thank you very much.
Thank you very much. Steve, do you mind to come to the stage, please? Now we will take a question from the audience. Maria, is it realistic to think that one day cancer will be eliminated, or do we need to learn to live with it?
Well, cancer is a disease associated with aging. So as long as we are aging, I think we will have cancer. So in fact, the prevalence of cancer is increasing, the incidence of cancer is increasing owing to demographic aging. So the prediction is that in 2050, the number of cancer cases will be almost double than now.
Wow.
Having said this, we have every year we have new treatments for cancer, and we have an increased survival of cancer. But cancer is a disease which is associated to aging. So as long as we age, we will have cancer.
Thank you. Steve, will it ever possible to calculate the exact biological age of a person?
Yeah, the word exact is a stumbling block. So it's, we can approximate it, but there will always be a big variation, you know. So imagine somebody sleeps very poorly, you know, then it affects in certain ways their state, you know. But then 2 weeks later they go on a vacation, then they're well rested, you know. So many measures such as glucose, for example, will pick up these changes in sleeping patterns, you know. And also conceptually, we of course work on epigenetic clocks, but they really give you only part of the story. You know, you want to measure so many other biomarkers. Maria mentioned, for example, telomeres, you know, or other markers of senescence. So yeah, I think students will continue to write doctoral dissertations for many, many years on this topic of biologic age estimators, you know. I always think of it like different versions of telephones. We already have biomarkers that are remarkably powerful, you know. And so yes, we can do clinical trials of anti-aging interventions, but I think every year we will have ever better biomarkers. Yeah. Mm.
Thanks. Maria, up to what point are aging and cancer linked to the current studies?
Well, I think there is a very clear link because cancer is an age-related disease. So cancer happens because our cells accumulate DNA damage. These damaged cells can either cause a degenerative disease because of the lack of regeneration of a tissue, or these cells can become immortal. Usually they do that by activating telomerase and maintaining telomeres, and they could give rise to, or they could give rise to a tumor because well, telomerase is not leading to the tumor. This is done by the loss of tumor suppressor genes, or is done by oncogenes, but it's allowing for the growth of these cancer cells.
Thank you. And the last question to Steve. How can epigenetic clocks help to prevent diseases?
They help because they help you identify drugs that slow aging. That's really the way. I mean, many people measure their epigenetic age because there are several companies that offer this test, but I don't think this marker is very useful at this point. And the reason is we don't have interventions that reverse epigenetic age, or at least we don't have validated interventions, you know. So at this point, these biomarkers should not be measured as part of your annual physical. But the hope is, of course, that at some point we will have drugs, and then these biomarkers could become valuable to monitor treatment success, you know. So basically they could tell you, increase your dose of this pill or lower the dose. Yeah. Mm-hmm. Mm.
Thank you so much. And thank you, the audience. This is the last session of this first Age Open Science. We hope we solve you of the question about longevity, and then you enjoy the visit to Salamanca and some of the most important researchers about longevity on this field in Salamanca. Now, I would like to call to the stage Oscar González Benito, Director of Fundación General de la Universidad de Salamanca.
Muchas gracias.
Gracias.
Buenos días a todos, bueno, buenas tardes ya. Muchas gracias por el interés mostrado en nuestras jornadas. Muchas gracias por tolerar estoicamente las inclemencias climáticas. Nuestra preocupación cuando organizábamos el evento era el impacto que podría tener la lluvia. Al final ha sucedido todo lo contrario. Hemos tenido un exceso de sol, pero bueno, esto nos ha impedido cumplir nuestro objetivo. Hemos tenido aquí la élite de la investigación, los mejores investigadores a nivel mundial en materia de envejecimiento y de longevidad. Y hemos querido transmitir todo su conocimiento, hacerlo cercano a la sociedad, hacerlo cercano a los ciudadanos y al fin y al cabo prepararnos para todos los desafíos que conlleva esto, que es un éxito social como es la longevidad, ¿no? El incremento de la esperanza de vida. Solo me queda agradecer muy sinceramente a los investigadores invitados su participación en este evento. Como han podido constatar, son investigadores de primer nivel internacional, sus agendas son complicadas y aún así han encontrado espacio en sus apretadas agendas para poder colaborar con nosotros. Un agradecimiento muy especial al doctor Juan Carlos Pisúa, que nos ha ayudado desde el primer momento y que ha sido clave para que esto sea una realidad, y también un agradecimiento a los investigadores de la Universidad de Salamanca, los investigadores locales que también nos han ayudado y han participado en los eventos. Muchas gracias. Desde el Centro Internacional sobre Envejecimiento, nuestro CENIE, promovido desde la Fundación General de la Universidad de Salamanca, vamos a seguir trabajando en todos esos retos y todas esas oportunidades que conlleva el envejecimiento y la longevidad, y también vamos a intentar que esto no sea un evento puntual, sino que estas jornadas de acercamiento entre ciencia y sociedad se repitan en el futuro, y ya estamos trabajando en una próxima edición. Muchísimas gracias a todos y que tengan un buen día.